Well, hey Anatomy, hope you're doing well. When you watch this video, this is going to be part one of the immune system. Remember, I do have questions in this video that I would like for you to answer and email to me. Um, and hopefully I'll be helpful and clear in this video. Again, hope you and your family are doing well when watching. So the immune system, this picture right here is actually a picture of a white blood cell attacking and engulfing a bacterium or multiple bacteria. And so the immune system protects us, protects our body from pathological. Pathogen means beginning of suffering. Pathos means suffering. And so bacteria, foreign tissue cells, and even cancerous cells. Non-specific immunity. So the first one we're going to talk about, there's two different types of immunity. Non-specific and specific, or innate and adaptive. So non-specific is also called innate immunity. You need to know these vocabulary terms because it does not require prior exposure to an antigen or a signaling molecule on the outside of certain cells or you know bacteria. Those are still cells, but antigens on the outside of cells. So skin. So these are all examples of non-specific immunity. That skin's just a barrier, right? Tears and mucus, cilia, stomach acid. I guarantee probably every time we eat a meal, we're eating some sort of bacteria. We're eating something that's probably not good for us to eat, but stomach acid takes care of that. And we will definitely talk about inflammation, okay? So here's a table that I would probably pause and kind of look over or at least know where it is. But non-specific is also called innate immunity. Let me try to draw here on the screen if I can. So non-specific, innate immunity, and then specific immunity is called adaptive. So in this video, I'm going to be really talking about in non-specific immunity or innate immunity, okay? And so with that being said, let's look at another graphic here. So innate immunity, recognition of traits shared by broad range of pathogens. This is a rapid response. So this is just our general barriers, right? Barriers, skin, mucus, secretions. We actually will also talk about internal defenses, such as phagocytic cells, cells that engulf. Remember that first picture I showed you. Natural killer cells. We'll talk more about these in this video. Antimicrobial proteins and the inflammatory response. I'm not going to talk about adaptive in this video, but I will in my next video, part two. So some of the objectives for today. I want to help you understand the overall components of innate immunity, non-specific. I want to explain how physical barriers, what role they play, what role phagocytes play, what role natural killer cells play. I'll briefly talk to you about interferons, and that's really more of a college discussion, so we're not going to get into that in high school. Complement activation. The complement system is very important. And then finally, inflammation and fever, which we've all probably have dealt with. So first off, immunity. There's two mechanisms of immunity, innate and adaptive. Innate, physical barriers. It contains, this is just our general defense system. So physical barriers, phagocytes, our immune surveillance, constantly watching out for abnormal cells, natural killer cells do that. Interferons help us deal with viruses. We all know about viruses in this day and age, right? Complement is proteins that help assist antibodies, which antibodies label things for destruction, inflammation, and fever. So you need to understand all of these different roles, these players in innate immunity, and that's my goal for this lesson. And so that's another same of the slide. And then so this is adaptive. Adaptive, which I'll talk more about in part two, but just a brief little introduction is it's specific. And we'll talk about vaccines and specific, memory cells and various things that attack specific, dangers that we come into contact with. So here's a good picture that we've already talked about, but these are all of the, non-specific tools that we have in our bodies to help us with immunity. So questions, so here's your questions of part one. Distinguish between innate immunity and adaptive immunity. Tell me the differences between them. How does innate immunity protect us from disease? A child falls off her bike, happens to me a lot, and skins her knee. Which form of immunity will be activated immediately? They're talking about innate or adaptive. And then finally, this picture, what's this picture? Well, this is flesh-eating infection. Necrotizing fasciitis, I believe. Vibrio bonificus is a bacterial species that actually live in the coastal waters of Gulf of Mexico. And so I want you to do a little research for me on this. And by the way, these questions will be worth way more than 100 points. I'll probably make them worth like 250, 300. You know, I want you to do good work with these. Where is it found? What does it cause? How can we prevent ourselves from getting infected by it? And then I gave you some resources here. I'll put this link in the description if you want to click on. So that's questions part one. Not only do we see general barriers, but we also have help from the integumentary system, which we've already covered. So the skin gives us that physical barrier to the outside world. A lot of us like to wear gloves, but if you don't have any cuts or anything on your fingers, like your skin's a great barrier. But what happens and what you'll find out with that flesh-eating infection is that if there are cuts or if there's wounds, that can be very dangerous because your barrier is like a hole in the wall. Like remember the wall in the famous wall in the Old Testament, Jericho, right? That thing was well fortified. But what if there was like a weakness in it? Well, it's no longer a fortified wall. It's no longer a complete defense system. Your skin acts in a similar way. And so from sebaceous and sweat glands, oil glands and sweat glands wash away microorganisms. They also contain chemicals that kill bacteria, destructive enzymes called lysozymes, and antibodies. The integumentary system also hair. Hair protects us from physical abrasion and prevents hazardous materials or insects from getting to the skin. Stratify, which means multiple layers, right, gives us those multiple layers of protection. But not only does the skin and hair, but also other epithelial liner that is, involved in linings that are open to the outside world. And this is where mucus comes into play. So it's found along, remember, the digestive system, respiratory system, urinary system, reproductive system. And these cells also provide a physical barrier, but more than that, these secretions protect us. The mucus, the enzymes, even stomach acids often ensnare, destroy, or wash away pathogenic material. And so questions part two, how does the integumentary system protect the body from pathogens and possible infectious organisms? Okay, so I want you to kind of talk to me a little bit about that in a couple of sentences. Phagocytes, it literally means eat, cells that eat. And so they engulf and destroy foreign substances. You can see here that this is a phagocyte engulfing bacterial species, circular bacterial species, might be Staphylococcus, don't know. But first line of cellular defense, it can attack or remove microorganisms even before lymphocytes detect their presence. So these guys are really our first line of cellular defense. We're not talking about physical barriers here. This is when something actually gets past the physical barriers. We're talking about cellular defense with phagocytes. There's different types that they target, but all function in basically the same way. So here's examples of phagocytes. We'll go into this more when we talk about the circulatory system and blood cells, white blood cells and red blood cells. But for our purposes right now, phagocytes, there's first off neutrophils. They're very fast acting and mobile, and they clean up cellular debris or bacteria. Eosinophils, which are less abundant than neutrophils, they phagocytize foreign compounds and antibody-coated pathogens, so pathogens that have been targeted for destruction. Eosinophils take care of that. And then phagocytes also, there's macrophages that come from monocytes, which also help to clean up the system. So here's a picture here about the different types of phagocytes. It's kind of what they look like. And there's fixed ones and free ones. Free ones, of course, are roaming. But how do they know what to attack? Well, phagocytes can maybe attracted or repelled by certain chemicals. Right here is very important on this slide. Certain chemicals. So this is called chemotaxis, the movement toward or away from a certain chemical substance in the environment. They're sensitive to chemicals released by body cells or pathogens. It always begins with adhesion. They have to stick to the cell, to its target cell in order to eat it or engulf it. After adhesion, the phagocyte may either destroy the target or promote its destruction by activating specific defenses. So questions part three, let me take off the writing here. Questions part three. I want you to define chemotaxis, which I literally just did for you. And then what are some ways pathogens, bacteria and viruses, avoid chemotaxis? So of course, we've dealt with infections, viral infections, common cold, maybe bacterial infections, strep throat maybe. How did they slip past those pathogens, those defenses, those pathogens? How did they slip past our defenses? So the immune surveillance, constant monitoring of normal tissues. This happens by natural killer cells. So normal cells are generally ignored by the immune system. Cancer cells, however, have different antigens, different molecules on the outside of their membranes. And so natural killer cells actually recognize these as abnormal and destroy them. Remember, your body knows what you need to look like on the outside of cells and knows when it's not you. And it doesn't play around. It's not like, oh, let me sit down with coffee and investigate if you're dangerous or not. It just destroys it. It just goes for it. And so natural killer cells recognize all these foreign cells and destroy them. How does this actually work? Well, here's your natural killer cell here, and here's the abnormal cell here. But you see antigens, those molecules on the outside, they're different than your other cells. And so the NK cell recognizes this, binds to it. Then the Golgi, remember biology, right? All those fancy, fun ways to remember the organelles, ribosomes, Golgi apparatus, rough ER, smooth ER, lysosomes, all that stuff. Some of you are like, stop, the memories are too much to handle. But the Golgi actually moves towards where the natural killer cell has adhered to the target cell. The Golgi then starts secreting perforins. These are actually molecules, and we can see right here, that create pores inside of the cell membrane of the target cell. And this is exactly how NK cells destroy the cell, because once there's pores in the cell membrane, that cell can no longer maintain homeostasis, and therefore it slowly is, or maybe quickly, destroyed. It disintegrates, and that's called lysis. It is broken up. So questions part 4, how do natural killer cells detect cancer cells? And then I want you to do a little research on the second question here, but if natural killer cells are engaged in immune surveillance, how do cancer cells spread then? Because we've known probably someone whose cancer spread, tragically. This is obviously dangerous, and sometimes healing can happen. Sometimes God decides to take that person home, hopefully if they're a Christian. But if natural killer cells are always monitoring this stuff, how does cancer cells evade that surveillance? I want you to do a little, research with that and answer that for me. Okay, moving on, we have interferons. Small proteins, what are they? Small proteins released by activated lymphocytes. We talked about lymphocytes, T cells, B cells, macrophages, and virus-infected tissues. They trigger production of antiviral proteins in cytoplasm of nearby cells. They also stimulate activities of macrophages and natural killer cells. So you can see how important these proteins would be if we're dealing with a viral infection. Examples of cytokines. So chemicals, what are cytokines? Cytokines also help with our nonspecific immunity, chemicals released by cells to coordinate local activities. So a system that really helps you have these just general nonspecific immune responses is the complement system. The complement system is a part of the immune system that, hence the word, enhances or complements the ability of antibodies and phagocytic cells to clear microbes and damaged cells from an organism. They actually promote inflammation. Inflammation is meant to help us and heal us, but sometimes it doesn't, tragically. And then attacks the pathogen's cell membrane. So I want to show you a picture here. Here's a picture that I got from another textbook, but this picture is from Pearson. It's probably a little bit more clear. Let me take off the writing here. But how does this actually work? And there's different pathways that the complement system works, but for our purposes, let's just deal with the classical pathway. So you have antibodies. So this bacterial cell has been labeled by our system with antibodies, like targeted for destruction label. What happens is that a certain complement protein attaches to these antibodies, but a certain part of it is cleaved off when it attaches to more than one antibody. And we'll consider that C3. C3 diffuses away. So that's the picture here, right? C3 diffuses away. And so you can see that causes a signaling cascade. A signaling cascade is basically like something happens in one place of the cell and it triggers this enormous response by activating something different and then activating something different. And so one thing activates 10 things and those 10 things activate 1,000 things. And so it's literally like a trickle of water eventually becomes a roaring waterfall. If you ever played dominoes, right? You knock over one domino, but if you've seen like those amazing videos where people like build art with dominoes, like knocking over one domino could knock over 1,000,000 dominoes if you arrange them correctly. And so this is what a signaling cascade is. And so what literally it does is that it causes all these molecules to bind to bacterial surfaces and enhances phagocytosis. So it enhances the immune system being able to engulf these particles and destroy them. All pathways split inactive C3 proteins to form activated C3b and C3a proteins. And all these proteins are doing is that they're increasing the chance of the immune system to destroy these cells either through phagocytosis or lysis, which is causing, there you go, there's those holes in the cell membrane, multiple pores in bacterium, the cell is destroyed. Okay, so that's an example of that. So the last thing that we're going to talk about, and you can see the magnifying glass here, is inflammation. There's an amazing story about penicillin. I'm not sure if you're allergic to penicillin. Penicillin was one of the first antibiotic to be discovered by Alexander Fleming. Penicillin, I'm allergic to it. At least I was told when I was young. I probably need to get tested for it again. But penicillin, the discovery of it revolutionized modern medicine. There's a story about a gentleman, a constable, a police officer, and he got either prick. I forgot if it's on the hand or the face. I think there's different stories about it, but I want you to research, and I'll talk to you about this later on. But Albert Alexander, and I'll put that link in the description as well. But Albert Alexander, he was actually the first patient of penicillin, and I want you to kind of read about that because we live in an antibiotic world. We have antibiotics for everything. We give antibiotics to livestock, or at least we used to before everybody started to expose that danger. But we take antibiotics, all the time. Doctors prescribe antibiotics. Some of them prescribe very quickly, but that wasn't always the case. Sometimes antibiotics, 1920s, 1930s, antibiotics weren't around. And so what happened when you got an infection? Well, you either survived, and many people determine that lucky, although a Christian doesn't believe in luck. But sometimes you didn't survive, and you developed something called blood poisoning. Blood poisoning is just a way of saying the infection is spreading. The bacterial cells are invading the blood system. This can lead to something called sepsis. Sepsis occurs when chemicals released in the bloodstream to fight an infection trigger inflammation throughout the entire body. So if the bacteria is spreading through the bloodstream, then inflammation is also spreading possibly, and you're maybe not going to make it, especially if you don't have antibiotics around. So what exactly does inflammation do for the body? I'll take away the webcam here to help you understand, see this feedback loop. But when tissue injury is caused by physical or chemical agent or microorganism, the capillaries widen, there's increased permeability, which means there's leakage, increased ability for cells to get where they need to go, attraction of white blood cells, and a systemic response. So what happens is that you have these symptoms: heat, redness, swelling, tenderness, and pain. I'm sure you've all kind of felt these symptoms when you've been inflamed, whether it be from a cut or an injury, or maybe you just kind of feel this way internally sometimes. And I'll talk to you about chronic inflammation. On the right side of the screen, this is considered a feedback loop. You can kind of read through how this exactly works, but healthy tissue and bacteria enter the tissue damage occurs, and then inflammation begins. And then you can kind of see the tissue repair back to healthy tissue. Sometimes additional things need to be activated. And I'll just kind of read through that feedback loop to help understand the inflammatory response. But it's brought about by molecules released upon injury. A very important cell here that you should recognize are something called mast cells. Mast cells release histamine. If you take allergy stuff, some of those might be histamine blockers, which triggers blood vessels to dilate, which means become wider, and become more permeable, which means they become the ability to recruit more blood flow, increase blood flow. Activated macrophages and neutrophils release these cytokines. Cytokines are signaling molecules that enhance the immune response. So inflammation is designed to protect us from pathogens. So here's a picture here. Let's say you get stuck with like a needle or a pen or even a piece of a splinter from wood, and it causes damage to the connective tissue or fibers or into the tissue. Causes a change in chemical composition of the interstitial fluid, remember, between the cells. Well, these chemical composition, your body can sense that, and so it causes a change in that. Damaged cells release prostaglandins and proteins and various ions. Foreign proteins or pathogens may have been introduced as well. So it triggers a complex inflammation response, and these mast cells are activated due to that environmental change. Something that's also a result of inflammation is pus. White blood cells, dead pathogens, cell debris. You can see that this is pus from like a zit type thing. But all of that is leftovers from fighting a difference in environmental chemical compositions. All of this they were brought to a site to defeat maybe bacteria, maybe inflammation, maybe tissue damage. And so here's just another picture here. So you have your splinter, bacteria is on the splinter. It causes mast cells to release signaling molecules like histamine, macrophages releasing their molecules. It causes, and those molecules, signaling molecules, right? So they carry a signal and they cause change wherever they go. And so they cause changes in the blood vessels. You can see that the blood vessels are now more permeable, which white blood cells can now escape and go attack. They're doing chemotaxis to do that. Phagocytosis is also helping it. So we're trying to clean up that wound. Redness might occur next to the wound. Why is that? Because the red blood cell and the capillaries being wider and all these cells moving into that situation to help defend our bodies. And so this picture right here just kind of walks you through what I've been talking about. If you want to pause it, you can and kind of look over that. The inflammation response can either be local or systemic. So it can be like localized, but it actually can be a whole body response. And so fever is an example of a systemic inflammatory response triggered by pyrogens released by macrophages and by toxins from pathogens. We already talked about septic shock, but that's caused when an overwhelming inflammatory response. This video right here is, I think, about septic shock, but you don't have to watch it. So the fever, body temperatures, anything greater than 99 degrees is, I guess, considered a fever. But, most people don't, above 100, roughly. But I'm not a doctor, of course, if you're not feeling well. But pyrogens, fever, heat, and gin means substance or beginning. So they begin a fever. They're fever-inducing proteins. They reset the temperature thermostat in the hypothalamus. The hypothalamus controls our temperature, and we range, 98.6, but that's not, we have a range. Like we sometimes are low, sometimes are higher. So, usually a fever is, I'm gonna have to look up 99 degrees. It seems a little low from what I've been told by doctors, but nonetheless, of course, when we get 102, 103, 104, that is really where we start, okay, this is a really high fever. This is concerning, of course, fever and COVID-19, one of the symptoms of COVID-19. But it can be beneficial. See, some doctors actually are like, listen, the fever's, 101, 100, they might be like, don't take anything, let it run its course. It's actually helping you. And that's obviously easier said than done because the fever doesn't make us feel very good, but it can be beneficial within limits. It may inhibit some viruses and bacteria. It might be making them hard to replicate or move or grow. It also might increase the metabolic rate, which might speed up repair processes. So the last question, this is the last slide for this video, is questions part 5. How was penicillin discovered, and what is Albert Alexander's role in penicillin's discovery? So I want you to do a little research on that. Talk to me about how penicillin was discovered by Alexander Fleming and Albert Alexander's role in penicillin's discovery. Chronic inflammation could be affecting millions of people. I don't know if you've heard about it, but chronic, if you might be chronically inflamed. What are some diseases or foods or other environmental factors? See, some foods cause inflammation in the body that could cause chronic inflammation. So tell me about 3 to 5 different diseases and or foods that could cause chronic inflammation. Of course, long-term inflammation would be hazardous to our health. It would cause a lot of different symptoms and changes in daily lifestyle and how we feel. Finally, how exactly does the hypothalamus cause a fever? I have no idea how JK is right there. So let me erase that. I'm just, maybe that's oh yeah, Ella Grace came in and started typing. So anyways, yeah, that's not just kidding. I do want you to answer that question. But how exactly does the hypothalamus cause a fever? What exactly does it do to increase, obviously there's those proteins involved, but what do those proteins actually do in the hypothalamus to cause it to reset the thermostat and actually crank up the heat in our bodies? So until next time, guys, I hope this video was helpful. If you have any questions, reach out to me. Until I guess we talk or see each other again, God bless. I hope you are well. Get in God's word. There's hope and peace and Him to be found in it.